All right, everyone, welcome back. Welcome back for the Ignite Talk portion of the afternoon. I hope you enjoyed the poster session and all of you have voted or at least have voting ballots to submit for the best poster and best Ignite Talk. We had plenty of posters out there. If you did not have a chance to look at all of them, they'll be up for the remaining of, I believe next week. Maybe even longer, and we'll have those up for everyone to peruse. And I just want to throw this out to all of our summer interns who are here in the audience to take note that these are posters that would be similar to the posters that you would be presenting theoretically after this summer at also some of the scientific meetings regionally or nationally. Just a reminder that the awards for best poster is $100 and best Ignite Talk is $200. So please fill out a ballot and put it in the ballot box out there. So today's oral presentation format is called an Ignite Talk, which will be a short, fast-paced talk from students about their research and why it's important. This year we had 12 graduate students who are presenting from OSU's Comparative Health Sciences Department, the Fisheries and Wildlife and Conservation Sciences Department, Department of Integrative Biology, and Microbiology. I'd like to invite our first speaker, Spencer Luna.
Spencer is unable to be here today, so he has sent us a video. So we're going to play a video.
Hi everyone, my name is Spencer Lunda. I'm a third-year PhD student at Hatfield Marine Science Center at Oregon State University, and today I'll be talking about some research that my colleagues and I conducted on the improvement of Pacific oyster settlement using probiotic-treated substrates. So, the Pacific oyster industry. The West Coast contributes about a third of total U.S. shellfish production. In 2018, this was $106 million, so a pretty substantial industry, and farms that grow the oysters are dependent on the production of these larvae on the left from hatcheries to produce the oysters. An issue with that, though, is that many larvae in a hatchery environment will never actually settle and metamorphose into a spat or a juvenile larvae, or a juvenile oyster, rather. So these are some controls from an experiment I did, and you can see that metamorphosis rates were never greater than 30%. So as a solution, how can you improve settlement of these larvae? So marine bacteria that provide benefits in growth and survival to oyster larvae when exposed to a pathogen, those are the probiotics that you can see on the plate here. If those improve growth, maybe they also improve settlement. And why would that be? So bacteria could be forming a biofilm on the surface that the larvae will choose to settle on, and they could be generating a chemical cue that attracts the larvae to that surface. And some known cues are GABA on the left and ammonia on the right. So to test this, we wanted to do some experiments with soaking these tiles in liquid probiotic cultures. So soaking the tiles while the bacteria are growing, maybe they'll form a biofilm on that tile, and then we can use those tiles to test if there's settlement, improved settlement on those. So how do we do this? We did some well plate trials to start out. So those were using these small ceramic tiles here in the picture. They were soaked in the liquid probiotic culture for 24 to 48 hours and then transferred to a well plate. So here's the well plate. So each tile was placed in the wells, and then seawater and settlement-ready larvae were added to each well. I also tested marble tiles to compare to ceramic to see if there was comparable settlement on those two surfaces. And we can see the results of that here. So generally speaking, marble and ceramic tiles produce comparable settlement, as can be seen between these comparisons on in each section of the graph, which was encouraging because the marble or ceramic tiles are more available. So moving forward, we wanted to set up a tile assay and test all of these different combinations of probiotics. So each of these letter combinations you see at the bottom is a different either single probiotic strain or multiple probiotic strains, and you can see this is comparing settlement to the larvae-only control on the left. And same here, this is another graph. This was done with 48 hours of soaking the tiles in probiotics as opposed to 24 on the previous. And again, we see settlement was either comparable or maybe slightly improved for many of these treatments in comparison to the leftmost graph of the larvae control. But the issue with this was I didn't actually have much significance with my results. So moving forward, I wanted to try maybe scaling up. Maybe we'll get more significance with more larvae and more chances to observe a difference. So I decided to be culturing the larvae in 10 buckets instead, which you can see here. So basically how this worked was each bucket got 2,000 larvae added to it that were ready to settle with some autoclaved or filtered seawater, I should say. And then I added the soaked tiles or that were soaked in either seawater or the probiotic culture for each one. So this is what that looks like. On the left, in that yellowish liquid, that's the bacterial culture. So there's actively growing bacteria in there, and the tiles are there, and maybe maybe a biofilm is being formed there. And on the right, you see just the tiles soaked in seawater. So after soaking them, they're added and kind of stood up to allow a more surface area for the larvae to settle on. They sit in the buckets with the larvae for 72 hours, and that's just in the dark, and that's because larvae prefer to settle when there's no light. So after doing that, this is what the results looked like. So as you can see, there was little difference. Unfortunately, not quite what I was looking for, but maybe that provides some insights into the fact that the probiotic combinations I was testing here are not particularly effective for inducing settlement actively through a biofilm. Similarly, again here, this was a second treatment I tried, or a second tile trial I tried, and comparable results again between control and PV, so that stands for the probiotic treatment. And so you can see this is for settlement on tiles, settlement in the buckets themselves, and then combined is the total on the right. This is what those look like. Here is the tile covered in those little black specks. Those are spat or juvenile oysters that have settled. So you can also sometimes observe a pattern, and that might be due to settlement on a biofilm from a certain bacteria that formed there. So moving forward, we would like to first test some different combinations of probiotics that we have in our library and see what might be a preferable alternative. And then after that, we wanted to also look at DNA sampling of the biofilm to determine which strains of bacteria are present. And lastly, I'd just like to thank everybody for listening. This funding for this research was provided by 2020 Louis and Maud Hill Coastal Marine Studies Award, which I'm very appreciative of. And I wish I could be there. If you have any questions, let me know. Thanks.
So one thing that you can imagine coming back from 2 years of COVID and having the Markham Symposium in this wonderful auditorium that we have, this being our first Markham Symposium in person since then. Last year we had a virtual Markham Symposium, and at the height of the pandemic in the first summer, we did not have a Markham Symposium. So you can imagine the many challenges for not only our students to do their research and their mentors to advise them, but all the labs to stay open to support that research. And then the challenges of all of you, particularly the graduate students here, to try to continue that research and also present. And what's unique about this presentation format, and just a little bit of history of this, is Janet Webster, who is our retired Gwynn librarian, came up with the idea of why not have students do another form of communication. And typically what we see are 15-minute oral presentations that you would see at a conference or a meeting, 12 to 13 minutes of research, and then a couple more minutes of Q&A. What's unique about this Ignite talk path, or this way of communicating, is it forces the communicator to not rely on this, but to rely on this, right? How I can communicate to you individually and tell you what it is that's so incredibly important about the research I do. Why should you care? And do that in 5 minutes. So the slides are really a secondary item. And what's challenging to do this in this type of format is to do it in a format that requires you to record that without seeing the person. And Spencer does amazing research, which is why he received that award. So we do have a couple more videos, but I do want to highlight that Spencer Lunda is a PhD student in the comparative health sciences. His advisor is Carla Schubiger. And just to give you a little bit of background on the Lewis and Maud Hill Coastal Marine Studies Award, It's intended to support a graduate student pursuing research in coastal marine studies that include, but not limited to, aquaculture, which Spencer just demonstrated, oceanography, marine biology, and marine engineering. Okay. Unfortunately, Spencer isn't here to answer any questions, and we have a second video, Tatiana Galvin, who is a bachelor student, an undergrad in marine biology, in molecular biology, whose advisor is Rebecca Vega Thurber, and her award is the Holt Award, the Curtis and Isabella Holt Education Fund, and that is intended to foster education in the marine sciences by providing financial support to undergraduate or graduate students pursuing marine science studies. All right.
And for folks online that were having difficulty hearing, we're going to try this different format and we'll give it a go. Hang in there with us.
Hi, I'm Tatiana Galvin. Today I'm going to be presenting our research on microbial community shifts and disease scombers during our work on the Puffy Snout project. This research was conducted through the collaboration with the Monterey Bay Aquarium and the Vega Thurber Lab. We also worked with experts from the OSU Electron Microscopy Facility and the OSU Center for Quantitative Life Sciences. The Puffy Snout project is a collaborative investigation into the cause of Puffy Snout syndrome, a disease that threatens both aquaculture and aquaria and can jeopardize conservation efforts. So, what is Puffy Snout syndrome? It's a captivity-induced disease in scombered fish, characterized by the inflammation of and tumor-like growths on the tissue in the snout, as seen in the picture. As the disease progresses, feeding success declines and there are periods of abnormal swimming behavior, such as listing to one side and pitching nose down. Progression of the disease eventually leads to death, and causal agents are currently unknown. Our first step was to observe the disease using transmission electron microscopy. This would give us a detailed view of the cellular effects of the disease, as well as help us identify a noticeable presence, or lack thereof, of possible pathogenic organisms. All samples were collected by the Monterey Bay Aquarium. There were 6 symptomatic and 3 asymptomatic aquaculture Pacific mackerel, as well as 4 wild Pacific mackerel. The tissue from one of the symptomatic fish did not correctly fix for electron microscopy and so was excluded from analysis. TEM imaging was done at the OSU Electron Microscopy Facility at multiple magnifications, and cellular features indicative of disease were recorded, and notable features were further investigated and recorded. Here we can see some of the viral particles we found in the afflicted samples, indicated by the red arrows. And in image C in the lower left, we can even see some in what appear to be the process of budding. We also found swollen and damaged mitochondria in the aquaculture samples, as shown in the 2 images A and C, on the right, with the internal membrane and cristae often appearing entirely degraded or absent. So here's what we found. There were viral-like particles and severe mitochondrial and cellular degradation in the aquaculture samples, but not in the wild-caught samples, and no other pathogens were identified in the aquaculture samples. Now we're working on sequencing the 16S ribosomal gene, a universal gene marker of bacteria that can help us confirm and compare the presence of bacterial microbial communities between samples and environments. There's also currently a metagenomics project being conducted. Again, samples were provided by the Monterey Bay Aquarium. PCR methods were used to amplify and isolate the 16S gene from previously extracted DNA. Once isolated and amplified, the samples were sent to the OSU Center for Quantitative Life Sciences to be sequenced. Samples are prepped and run through the first PCR reaction, then run through the gel electrophoresis to isolate the 16S gene. The isolated gene was then run through the second PCR reaction, correcting for low DNA density present in the samples. This is an image of one of the gels made using the first step PCR DNA, as well as positive and negative controls. The white arrow is pointing at the 16S gene to be extracted and amplified in the next step. After the second PCR, the samples were checked for contamination and cleaned up. Samples were then sent to the OSU Center for Quantitative Life Sciences to be sequenced, and the returning data was then analyzed. Very little bacterial DNA was present in the samples, and the concentrations were too low to be able to do further analysis on bacterial communities in the samples. However, this matches the TEM observations that there was a noticeable lack of non-viral organisms. A low concentration of bacterial DNA is reasonable considering the notable lack of bacterial presence observed in previous investigations. Further research needed to confirm this and further narrow down potential causes. However, a bacterial pathogen is unlikely. Further research is needed to discover the cause and why it's only present in captivity. One experiment planned is to culture saltwater cell lines. Previous studies have tried cell lines, however, they were not saltwater or tuna cell lines, which could affect the pathogen's ability to replicate. Images and information was used from Voorhees' 2015 Epidemiology of Puffy Snout Syndrome in Tuna. Information was also referenced from Nakamura's 1972 Development and Uses of Facilities for Studying Tuna Behavior. Thank you so much for listening.
Okay, and that was Tatya Galvin. Our next speaker is Victoria Quinessen, master's student in Fisheries, Wildlife, and Conservation Sciences, and advised by Will White. She received the 2020 Holt and the 2020 Markham Award. I mentioned and described the Holt Award, but I do want to mention and describe the Mamie L. Markham Endowment Award. This is one of our largest awards here offered at the Marine Science Center for our student researchers. It's intended to assist graduate or postdoctoral level researchers and research utilizing OSU's Hatfield Marine Science Center.
Thank you. Oh boy. I'm ready. Hi, my name is Vic Quinessen. I'm actually a second-year PhD student here in Will White's Fisheries, Oceanography, and Population Dynamics lab based out at Hatfield. I would like to say first that I'm actually going to be talking about sea turtle persistence in the face of climate change. And then I would actually like to acknowledge the fact that we are on the traditional homelands of the Kana tribe of the Siletz Indians. We white colonizers forcibly removed them from this land in the 1800s, but they are still alive today, and they are part of the Confederated Tribes of the Siletz Indians and the Confederated Tribes of the Grand Ronde. Then I would like to thank my PIs that are on this project. So Dr. Will White, my advisor, Dr. Mariana Fuentes from Florida State University, and Dr. Lisa Komorowski from UMass Amherst. So the modeler, the ecologist, and the geneticist. So what's actually the problem that's going on here? So there's a few different things that are combining to form one problem. So first, sea turtles do not have sex chromosomes like humans do. Their sex is actually determined by the temperature of the nest when they're eggs. Second, warmer nests produce more females. And third, global climate change is causing nesting beaches to get warmer and warmer. And so we're seeing a lot of places where hatchlings are being more and more feminized. And the question is this going to affect populations? When, how? It depends on how many males are actually needed to keep populations alive through time. Unfortunately, males like to stay in the water. They don't come onto land to nest like females do. And so it's harder to keep track of them. It's harder to count them. It's harder to figure out where they're going and when. And so we don't really know. I am behind. So what we're doing is we're going to this tiny little island off the coast of Brazil and studying a subpopulation of green turtles that nest there. So green turtles, coloniomydas, sometimes I just say greens. And it's that tiny little beach called Praia de Leão, which is Portuguese for Lion Beach. And we have boat surveys where we go out and we collect males and attach satellite tags. So that's that blue splotch on the picture all the way on the left. And then we also have transect surveys where we're trying to identify how many males are in the area and when, and what they're doing, you know, resting, copulating, swimming, all that fun stuff. And then once we figure out how many males are actually in the area, how many of them are actually contributing to the next generation? You know, is one male doing all the work while all the others are watching? Are they all reproducing evenly amongst each other? We don't know. So we're also taking genetic samples to try to figure out how many males the females are mating with, how many females the males are mating with, and also how they're contributing to each individual nest. So, you know, is there one male that's contributing to all the eggs? Are the males kind of contributing evenly between them? And so my contribution for this project, because all of us went out to collect data, was I did some power analyses to try to figure out, once we have this estimate of how many males are contributing, how confident are we in that estimate based on how many hatchlings we're sampling from the nest, how many males there are total, and that kind of contribution between males for each nest. So, you know, for example, you've got male, red, yellow, and green, and they're kind of contributing evenly. You can sample like a third of that nest, and you'll pretty consistently get at least one hatchling that was fathered by each male. But on the other hand, if you've got Mr. Green here, who's only fertilizing 2 eggs in the whole nest, you need a much higher sample size to successfully identify every single male that's contributing to that fertilization. And so that's kind of what's going on with the population now, but what's going to happen in the future, right? Climate change isn't here and then it's not going to get worse, right? It's going to get worse over time. And so my part of the project is going to be using population dynamics of this population and stepping forward in time to figure out how the population may or may not persist under different climate change scenarios and under different adaptation scenarios using information from this population in Brazil. So looking at nesting data and hatchling data, figuring out how many nests the females are laying, how many eggs are in each nest, how many of those eggs are actually successfully hatching and making it out into the ocean by the end of the season. Here are some gratuitous photos of sea turtle hatchlings because they're adorable and the best. And so these are the different kinds of scenarios that I'll be modeling, right? So what happens if nothing changes and the turtles kind of keep doing what they're doing and humans don't do anything? What happens if there's a genetic adaptation, right? What happens if warmer nests can still somehow produce some males? What happens if the males can change behaviorally and travel a lot more to mate with females? And then what if there are conservation measures that we can do to help lower the temperature of nests and help kind of synthetically produce more males for the population going forward into the future? And how does that affect their probability of persisting over time? That was the first time I released a sea turtle. I cried a lot. It was great. It was a beautiful moment. And I think the next slide is just my acknowledgements. So I'd like to thank the Holt Award and the Markham Award for giving me monies to help me do this work. My co-PIs again, my wonderful collaborators in Brazil who threw me the best birthday party I've had ever. And also my amazing grad student union, the Coalition of Graduate Employees, fighting hard to make sure that I earn a living wage. And with that, I would love to take any questions. Oh God, do I have to choose? Just the stand.
Hello.
I would mute it and then put it in and then unmute it. Because you gotta like shove it in there.
Okay. So just out of curiosity, so when you say modifying conditions, is that like actually creating like artificial nests to have lower temperatures, or is that going to be a more overall, like if we, or hopefully when we work to lower emissions and then reduce the impacts of climate change?
Oh, well, that would be like the best goal ever, would be to reduce the impacts of climate change on everything, of course. But in the short term, that's looking more like moving them to a place where you can control the temperature, or potentially adding more shading or moisture to the beach to lower the sand temperatures that way. My paper is kind of just look more at how many degrees you would need to lower it for there to be consistent increases in the number of males or increase in population persistence in general, and kind of looking at hours that you would have to spend doing that, like frequency every year or every other year, 20% of the nest, 100% of the nest. Like, what are the combinations of effort that you would have to put in to lower the temperature of the nest enough for the population to actually persist over time?
Gotcha. Thank you.
Sorry, that was long-winded. I'll be around. I'm here for the free food. I can repeat it, yeah. So the question is if there's a critical time period during incubation that affects the sex determination of the hatchlings. Yes, absolutely. It's the middle third of incubation. That's the part where sex is determined. So theoretically, yeah, you could let the sea turtle do her thing, and then you could come back a few weeks later, and then you can dig up the nest and, do what you need to do and bring it to the lab or whatnot. You don't have to take it immediately after it's been laid to affect the sex of the hatchlings that come out about 50 days later.
Okay, thank you. Wasn't that better live? Yeah, all right. So see our next speaker and awardee is Hillary Thalman. So Hillary is a PhD student in fisheries, wildlife, and conservation sciences, advised by Jessica Miller. She received the 2019 Markham Award. There you go.
All right, thank you, Yi-Chung. I am. All right, hi everybody. My name is Hillary Thalman, and I'm a PhD student here at Hatfield. And today I'm going to be talking to you about how juvenile Pacific cod diet composition changed during marine heat wave conditions in the Gulf of Alaska. So in recent years, the Gulf of Alaska has been impacted by 2 marine heat wave events. And this graph that I'm showing you here is sea surface temperature anomalies for the Gulf of Alaska. And those areas of really dark red that you see on the map are areas where the temperature was about 3 degrees Celsius warmer than normal. And this had big impacts to the ecosystem, including declines in seabirds and marine mammals, pretty substantial declines in forage fish, and declines in ground fish. And one species that was particularly impacted was the Pacific cod, which declined by 75% in abundance following these heat waves, and which led to the historic first time ever closure of the federal Gulf of Alaska cod fishery in 2020. And as you can imagine, this had big impacts to the Alaska ecosystem. Cod are important prey for a variety of Alaskan animals, and they're the second most important commercial fishery in Alaska. And so there's a couple areas in time in the first year of this fish's life where they're going to experience thermal variability. And one of those areas that we don't have a lot of knowledge about is the nursery phase. So as these fish hatch in the shelf waters of Alaska and then move inshore to these shallow nursery habitats, we don't really know how they're responding to this thermal stress. And so the focus of my dissertation is how thermal variability impacts these juvenile cod in these nurseries when temperatures are a lot higher than they normally will be. And specifically for today, I'm going to be talking to you about how juvenile Pacific cod diet composition changes in these really warm conditions as prey abundances might change as these waters warm, and cod might be growing and developing and selecting their prey differently as they experience these warm conditions. And to do this, we go up to Kodiak Island and we do beach seine surveys off the beaches there from 2006 to 2019 as part of the NOAA Alaska Fisheries Science Center survey program. And we bring those fish back to the lab at Hatfield, we dissect them out, and we look at what's inside their stomachs. And here I wanted to just highlight the 4 undergraduate student workers who helped me process all of this data during a global pandemic, which was quite the undertaking. And I also want to highlight that this is just one piece of a much larger project. I'm talking about diet composition today, but we're also looking at how nursery growth varies in heat waves and body condition varies in heat waves. But today for the diet composition, we're going to look at diet composition using non-metric multidimensional scaling ordinations, which are a good tool here to compress this highly dimensional diet data into only a few dimensions for analysis. So each of the dots that you see here is an individual cod stomach, and the further apart they are from each other, the more different the diets are. And so if we color code this with red being heat wave diets, blue being before the heat waves, and gray being the 2 years that happened between the 2 heat wave events, we can see that there is some separation in diet composition. Those blue diets before the heat waves are quite a bit separate in space from the red diets. We also notice that prey is bigger during the heat waves. Cod are consuming bigger prey like polychaetes and mysids as opposed to smaller prey like copepods before the heat waves. We also notice that cod are bigger during the heat waves. Cod size is well correlated with these diet patterns, and the bigger cod are consuming the bigger prey in those warmer conditions. And so if we take a step back and just think about how the Pacific cod's size varies through time during these heat wave conditions, we can see that standard length through time is actually higher during these heat waves. So here is our 15-year time series in July and August, and we can see a big spike in growth in size in August. And so we know then now that Pacific cod are bigger during heat waves, and they're consuming bigger prey during those heat waves. And that can have implications for how well they survive their first winter and how well they recruit into the commercial fishery a little later in their lives. And there's a couple hypotheses for why cod might be bigger earlier in their lives. And in these stressful heat wave conditions, there's likely higher mortality at early life stages. And there's in fact evidence that in these heat wave conditions, cod eggs are only hatching in a narrow thermal window that was exceeded in the heat waves. So if there's fewer eggs, there's fewer larvae and juveniles, and the juveniles that survive tend to be the biggest and fastest growers. And I want to leave you with one other thought about why we might see changes in diet composition, and that is in these warm conditions, metabolic rate is likely also elevated, and that can impact diet choice as well. And if you didn't get a chance to see my poster a little earlier, I encourage you to check it out after the talks. Thank you. And I can take any questions.
The cod larvae, where you're showing the average size, was that for like the young of the year, or was that larvae that were more than a year old?
The size graph that I showed, that's
Yeah.
the juvenile Pacific cod. So that's their first summer in the nursery in July and August. So July is right when they settle in the nursery. August is after they've had a month in the nursery. And you can see that big size jump in August during the heat waves.
So that was the distribution, right? Not necessarily just the average. So it's not just that the younger ones are getting killed off, and so the average gets jumped up because there's fewer smaller ones bringing it down.
We do think that's part of the puzzle, is that, fewer of these fish are surviving to August, and those survivors are likely going to be bigger.
Cool. Thank you.
We have some larvae data, but it's being processed by our postdoc and some of our techs in our lab. So I encourage you to check that out once it comes out. Yeah, we think that's one of the hypotheses for why they're eating bigger prey. So the question is gape width of the fish contributing to the size of the prey? If bigger fish have a bigger gape width, are they able to eat bigger prey? And we think yes, but the challenge with having the survey data is that we don't have a lot of overlap in these cooler years and in the warmer years of fish of the same sizes. So it's harder to isolate both the heat wave and the size of the fish. There's a lot of fish that are just bigger in the heat wave years that aren't that big in the cooler years.
So I was also curious in terms of those prey items that you were talking about, are there actually, how does the population of those prey items change through those heat waves too? Like, are there less abundant, smaller prey items, so they're just, being eaten less because they're less abundant? Or do they maintain the same, similar populations between warm and cold years?
Yeah, that's a really good question. So the question is, are we seeing changes in abundance of prey, and is that impacting the prey choice of the cod? And there is some switching of prey items. There was more of these smaller copepods during the heat wave conditions that were more indicative of warm conditions. But that was more on the open waters of the Gulf of Alaska. In these nursery habitats where there's a little less of those currents moving the prey along, we didn't see as much variability in prey distributions during the heat wave. But one of the challenges that I've had is that there's just not a lot of data out there, not a lot of consistent data through time. So there's actually starting a citizen scientist project in the bay where these fish come from to see, does the zooplankton change through time? And so we're really eager to get enough years of that to see, what is happening with the zooplankton.
I guess an interesting thing is that you said that during the heat waves, there's like a whole collapse of, like, not collapse, but like each species had a decreased loss. Could this be explained by like bottom up control? So maybe like something in the phytoplankton?
Yeah, that's a really good question. What caused the collapse of these fish and this ecosystem? And we think that there are both bottom up and top down processes happening. There actually seems to be more phytoplankton in the warmer years, fewer zooplankton, and then fewer of these forage fish. And then with the fewer forage fish, that led to declines in the mammals and the seabirds. So it seemed to be the big piece of the system that collapsed during these heat waves was the forage fish. And that led to a lot of other animals struggling because those species weren't there.
All right, we have a question from online. Is there any genetic evidence of evolution? Of selection? Is there any genetic evidence of selection?
Yeah, that's a really good question. And it ties in really nicely with some new work coming out of some of the labs up in Seattle, where there is a genetic break, or there seems to be a genetic break between the eastern and western stocks of Pacific cod. But we don't have enough evidence yet from these cod to know if genetics was playing a role. But there is some exciting new findings coming out about genetics potentially playing a role in which stocks are there at what times. So we're excited to find more out.
You. Our next speaker is Megan Wilson, which will also be a video. Megan is a PhD student in the Department of Integrative Biology. Her advisor is Dr. Sue Spinagle. She received the 2019 Markham Award and the 2021 ODFW Award. And just to give you a description of the ODFW Award, the Oregon Department of Fish and Wildlife Marine Reserve's Graduate Student Scholarship was intended to support students at Oregon State University undertaking applied research that informs our understanding of the ecological processes shaping near shore, less than 3 nautical miles, marine communities in the Oregon waters.
Hi, everyone, and thank you for attending my talk. I wish I could be speaking to you in person today, and I do hope I have the chance to meet you all throughout the summer, especially our visiting scientists and interns. Before beginning, I want to acknowledge that my research occurs on the traditional Siletz, Yaquina, and Coquille homelands. These people experienced horrible atrocities associated with the forced removal from their land by white settler colonialists. Today, living descendants are part of the Confederated Tribes of the Grand Ronde Community of Oregon, the Confederated Tribes of Siletz Indians, and the Coquille Indian Tribe. As a researcher who benefits from access to stolen land, it's my duty to understand the complex history of my study areas and advocate for the reallocation of land to its original inhabitants. You can look up whose land you're living and working on at native-land.ca. For today, I'd like to start off by setting the stage. Our work occurs in coastal Oregon, which is a part of the California Current, a highly productive eastern boundary current system that runs along the west coast of North America and sustains some of the world's largest fisheries. The California Current is characterized by strong upwelling. And upwelling is the process where the combination of wind and the rotation of the earth pushes surface waters offshore. Cold, nutrient-rich waters rise up from depth to replace those advected surface waters, fueling the bottom of the food chain. Many fishes in the Northern California Current have an inshore and often benthic adult stage. Larvae are released into the water column and are pelagic for weeks to months and must feed, grow, and navigate a cross-shelf journey to settle in nearshore environments. Therefore, many species synchronize reproduction with upwelling cycles to maximize the chance that offspring are placed in suitable conditions to avoid advection, maximize food availability, and avoid predation. This synchronization often results in a single annual pulsive recruitment like we observe in the rockfishes along our coast. However, another nearshore groundfish, cabezon, recruits nearly continuously throughout the recruitment season. Cabezon have a benthic adult stage that lay benthic eggs that incubate for approximately 2 weeks before they hatch into the noostin. They're larvae that are pelagic for 2 to 4 months. Juveniles shelter in nearshore kelp and tide pools before descending to the benthos to grow and reproduce. Standard monitoring units for the recruitment of fishes are Smurfs, like the one pictured below the purple fish, mimic recruitment habitat, and we use them to monitor the recruitment of cabezon and other nearshore groundfishes throughout the recruitment season. We also conduct tide pool collections to collect larger, older juveniles, but for the purpose of this talk, I'll just focus on those early juveniles collected by Smurfs. The The SMURF project is a multi-institution collaboration where we use freediving to collect juveniles bi-weekly throughout the recruitment season, which spans from April to September. We've been sampling now for 10 years, and the project is ongoing. And so far, we found that cabezon recruitment rate is variable interannually and within a recruitment season, indicated by different rates among 3 different cohorts. One that arrives early in the season around May, one that arrives in the middle of the season, and one that arrives towards the end around July through September. To better understand this phenomenon, we need to better understand the factors affecting recruitment and how they might vary between cohorts and among years. We hypothesized that cabezon exhibit a portfolio of early life histories, and just as you might think a diversified financial portfolio increases the chance of investment success, a portfolio of early life histories might maximize overall recruitment success by maximizing the chance of offspring overlap with suitable environmental conditions. And to test this hypothesis, we measured early life traits using otolith microstructure in each of these groups. So all teleost fishes have otoliths or ear stones that aid in balance and orientation. They're similar to tree rings in that material is deposited onto them daily in a concentric fashion, and thus they can be used as a sort of flight recorder that helps us understand the experiences of fishes throughout their early life. Looking at these early life traits, we found that the late cohort in green is growing significantly faster than the other cohorts in all 5 years studied. And this is important because past studies have shown that faster growth can increase overall survival and recruitment by decreasing the amount of time a fish spends in sensitive stages. For example, the faster that a young fish larvae that is a poor swimmer and feeder can grow into a juvenile, the less time it will spend exposed to predators like jellies. We also found that, as we might expect, faster growth translates into a larger size at age. The late cohort in green is consistently larger than the other cohorts. And this is important because many predators are limited by the size of their gate, or literally what they can fit in their mouths. Thus, being bigger is better simply because you're large enough to gain refuge from gate-limited predators. So, we've observed different recruitment rates between cohorts, and we now know that some cohorts are growing faster than others and achieving larger sizes than others, but we still don't know what oceanographic or ecological mechanisms are related to this. So my next steps for this project are to explore the roles of temperature and prey availability on larval and juvenile cabezon growth and feeding, as these are 2 well-known mechanisms. And for a little sneak peek, we do know that the cohorts are experiencing different thermal histories. The shaded boxes indicate the period of time each cohort spent in the water column, and we can see that the mid and late cohorts are experiencing warmer and more variable water temperatures than the early cohort. In addition, preliminary findings are indicating that larval cabezon have drastically different diets between years and may be exposed to different prey fields. So to wrap up, we found a lot of interesting patterns, but this work is certainly ongoing, and it could not have been done without generous support from the Hatfield Marine Science Center scholarships and awards. Please come chat with me about these findings and my current work this summer.
Okay. Our next speaker is Kelsey Sueka, who is a PhD student in Department of Integrative Biology, also advised by Dr. Sue Spinagle. She was the 2018 and 2020 Markham awardee, as well as the 2020 Jones Award. And the Jones Award, the Walter G. Jones Fisheries Development Memorial Award, was intended to encourage graduate work in subjects that contribute to fisheries development.
Hi everyone, thanks for sticking around to listen to my Ignite talk. I'm sorry I can't be there with you in person, but I'm excited to give you a quick synopsis of some of the work I did during my PhD, focused on the impact of upwelling on larval northern anchovy in the Northern California current. Fish are ecologically, economically, and recreationally important to coastal ecosystems. And because of their important role supporting coastal food webs and human livelihoods, there has been a lot of work trying to understand why fish populations vary so much from one year to the next. Most marine fishes experience extremely high rates of mortality during their early life history stages, with far-reaching consequences for adult population dynamics. Growth and survival through the larval phase is generally considered a bottleneck for fishery production, and identifying the factors influencing fish vital rates is a core goal of fisheries oceanographers. Foundational theories in this field suggest that growth and survival are dependent upon the ability to find food and avoid predation in physically and biologically heterogeneous environments. In the Northern California current, a lot of that environmental heterogeneity is driven by upwelling. And coastal wind-driven upwelling occurs when prevailing equatorward winds combine with the natural rotation of the Earth to drive surface waters offshore, allowing cooler, nutrient-rich water to rise into the euphotic zone. During summer, upwelling circulation drives the establishment of an upwelling front, where higher density upwelled waters meet lower density offshore waters. And local upwelling intensity affects the strength and position of this front, and as a result, the cross-shelf extent of those cool, productive upwelled waters is highly variable in space and time, which has implications for the physical and prey and predator environments experienced by larval fishes. So for this work, I examined how spatial variability in northern anchovy growth and the distributions of their prey and predators relates to the position of the upwelling front. So to best make those relationships between larval fish growth and the zooplankton distributions, prey and predator distributions, we couple net sampling and fine-scale plankton imaging on 2 summer research cruises in 2018 and 2019 on the Newport hydrographic line just off our coast. Larval growth was measured using otoliths, which are fish ear stones that record age and growth information similar to tree rings. We then couple these growth data with in situ imagery, and this collage gives you an idea of what some of those images look like on the right. So what were the conditions like during sampling? In 2018, upwelling was strong and persistent, while in 2019, we sampled following nearly a month-long relaxation event. As a result, the upwelling front and cross-shelf region of cold, productive upwelled waters extended substantially farther offshore in 2018 compared to 2019. And this spatial variability in upwelling contributed to spatial variability in anchovy growth. Here I'm showing the mean daily growth trajectories, which summarize anchovy growth over each day of life. We can see that offshore anchovy grew significantly faster than inshore and shelf break anchovy in 2018. Whereas the reverse occurred in 2019 when the upwelling front was restricted nearshore and inshore anchovy had the fastest growth. So now let's look at the zooplankton distributions to try to understand the drivers behind this spatial patterns in growth. The plots I'm going to present are focused on copepod concentrations relative to the position of the upwelling front each year, with the front noted by a red dotted line. I also include the position of the stationary shelf break as a reference point to visualize how the extent of upwelling impacts the cross-shelf distribution of taxa. The shelf break position is highlighted by this solid red line. We can see high copepod abundances were observed inshore and across the upwelling front, not decreasing until about 20 kilometers beyond the front throughout sampling. And because the distance between the upwelling front and the shelf break varied, the relative availability of copepod prey beyond the shelf break also varied. You can see that when upwelled waters protruded farther offshore in 2018, high abundances of copepod prey were found beyond the shelf break. But when upwelled waters were restricted nearshore in 2019, copepod abundances were low in that dark blue box off of the continental shelf. And these observed regions of high copepod availability align well with our finding that growth was substantially faster offshore in 2018, but inshore in 2019. So in quick summary, not all ocean environments were equal at promoting northern anchovy growth and presumably survival. Importantly, the regions that led to the highest growth differed between years and were related to the spatial extent of cool, productive upwelled waters. Following sustained upwelling in 2018, anchovy offshore grew significantly faster than those inshore. Copepods were more widely distributed between inshore and offshore environments, and fine-scale anchovy copepod overlap peaked beyond the shelf break. Conversely, when upwelled waters were restricted nearshore in 2019, copepod abundances and anchovy copepod overlap were substantially higher on the continental shelf compared to offshore, resulting in a significantly faster growth inshore than offshore that year. So I just want to thank everybody for the reason that I'm here today, all of my funding sources, especially the ones that I'm here to represent today, all of my Markham Awards, everything from Hatfield Marine Science Center. Thank you very much.
Okay, our next speaker is an awardee is Jeffrey Walker, who is a master's student in fisheries, wildlife, and conservation sciences. Jeffrey is mentored and advised by Dr. Kathleen O'Malley, received the 2021 and 2022 Walter Jones Award, and here he is, and the 2021 Markham Award.
All right, today I'll be talking about my research on the genetic connectivity of Pacific albacore tuna. And I'll start by giving a very brief overview of how Pacific albacore are managed and kind of what they are. And then I'll talk about how I'm using genetics to address some major uncertainties. So albacore tuna are highly migratory open ocean predators that are built for speed. Their warm blood and torpedo-shaped bodies give them the speed they need to chase down smaller fish. Of course, we often think of albacore as food for us, as it's also a tasty and protein-rich seafood. And being fun to catch and great to eat is the perfect recipe for a valuable fishery species. Albacore are around the 5th most valuable fishery species in Oregon and are caught throughout the Pacific Ocean in Japan, the United States, Taiwan, and many other countries. And the current managing system splits albacore into 2 stocks in the North and South Pacific Oceans, separated by the equator. And this decision is primarily based on the lack of albacore caught near the equator, as well as a lack of convincing tagging data showing migration across the equator. However, this tagging data is based on relatively low return rates. And furthermore, we're still missing lots of critical information on albacore distribution, including definitive spawning areas and migration routes. So a lot of the current information is being loosely approximated. And it can be really difficult to collect data on massive, highly migratory marine populations like albacore, which in turn hinders the development of effective management strategies. But remember, it's called a tuna can, not a tuna can't. And so with that spirit in mind, we can look to genetics to help us tackle some of these problems clarifying distribution. And recent research here in the State Fisheries Genomics Lab was able to distinguish North and South Pacific albacore using their genetic differences. And now I'll be looking at these differences to answer a few main questions. Specifically, my first objective is to try and see if there are any genetically distinct groups of albacore in the North or South Pacific Oceans. Second, I'm attempting to identify albacore that have migrated across the equator or that are hybrid offspring of North and South fish. Lastly, I'll be testing the accuracy of a potential genetic sex marker and investigating sex-specific distribution patterns. This is important because traditional sex identification is difficult and expensive, so a genetic marker would make collection of sex data much easier. In order to do this, I'll be looking at 289 target locations on the albacore genome, or genetic markers. And these 289 consist of some of the most informative markers from the previous study, as well as the genetic sex marker that I'll be evaluating. And using a smaller subset of markers allows us to include more fish in the study. From 2020 to early 2022, we coordinated a sampling effort throughout the Pacific Ocean by reaching out to collaborators to help collect tissue samples. I was also able to collect some fin clips myself right here in Newport as the fish were unloaded on the docks. And over 1,600 samples were collected in total, and most of these are from Oregon and Washington since they are the most accessible. But we aim to have sampling coverage on both sides of the North and South Pacific so that we'd be able to look at intra-hemisphere structure more clearly. After extracting DNA from each sample, I amplified the 289 target regions and attached a DNA barcode to each amplified fragment. So this unique barcode, which is made of DNA base pairs, actually tells us which fish the DNA came from so that we can trace it back later. And this allows us to pool DNA from many fish into a single tube so that we can sequence them all together in batches. And it still kind of blows my mind, but this small drop of liquid outlined in red actually contains all the genetic information we needed for nearly 1,000 fish. Once the information is sequenced, then I genotype each of the target markers, which creates a sort of genetic profile for each fish. And using this, we can determine if each fish originated from the North or South Pacific, and if the sex marker ends up being accurate, what sex it is as well. And using these genetic profiles, I'll then separate them into groups based on their genetic variation. So this is an example principal component analysis plot from the previous study that shows how the North Pacific fish in orange and the South Pacific fish in blue cluster into separate groups. I'll also use this and other analytical approaches to identify potential hybrid individuals, like you see in the gray cluster in the middle, as well as potential migrants between the North and South Pacific Oceans, which are clustered into the opposite group and are indicated here by arrows. And getting phenotyping sex data is difficult, so not all these samples will be used to measure sex marker accuracy, but early data is showing that our sex marker has an accuracy of about 76% in South Pacific fish, which is a lot lower than expected. We're hopeful that it will perform better in North Pacific fish. Through a better understanding of genetic connectivity in Pacific albacore, we can begin to clear up the fuzzy picture of their distribution. Furthermore, establishing accurate and efficient genetic sex identification will identify the collection of sex-specific parameters that are critical to stock assessments. And with that, I'd like to thank some of my sampling collaborators listed here. And of course, a special thank you to the Mamie Markham Research Award and Walter G. Jones Fisheries Development Award, which have been instrumental in funding research costs and are the reason I had the opportunity to present here today. Thank you for your time, and I'll take any questions.
Thanks for your talk. That was fantastic. I'm curious if you, in your research, have found more local populations than just between the general North Pacific and South Pacific, if you found more specific populations within those regions.
Yeah, so firstly, I do want to clarify that these areas that we're sampling are not necessarily populations, as we still don't know a lot about where the albacore spawn, but we are looking at genetically distinct areas or different groups. And currently, these have all been sent out for sequencing, but we haven't, we're starting to receive the data now. The last batch is coming in, so I haven't actually gotten to dig into it. But that's one of those questions that we're really interested in answering, is if there are any more distinct groups within the North and South Pacific. And particularly, we're looking at French Polynesia, as there's been some evidence before that it might be distinct from other South Pacific regions. But we don't know if that's from a different spawning site or if it's just similar to some other group.
Hi. So my question is on the I think the PCA plot you showed where you had the hybrid population or the hybrid group and then the migration. Is the migration because they're leaning more into that blue zone, or is it, what's the difference between hybrid versus the migration?
So the migrants are those that they're orange, so they were caught in the North Pacific, but when they cluster on the PCA plot, they cluster with the South Pacific fish. So that's kind of telling us that they're genetically similar to South Pacific fish, even though we sampled them in the North Pacific. So that's kind of an indicator that they might actually be migrants that were originated in the North Pacific before swimming to the South Pacific or vice versa. And the hybrids are ones that show genetic ancestry from both populations.
Can you tell us what inspired you to study tuna?
I mean, I think they're a really fascinating study species for one, just as they're highly migratory predator like that. I think a lot of us, you know, myself included, I ate them a lot growing up, and I just think of them in cans as little fish in the ocean, but they're really awesome fish, both for catching and just in general. They're one of the few warm-blooded fish that that have like warm blood, which helps them chase down those other fish. So I just think they're really neat study species. And we had some great research that I could springboard off of going on already in the lab.
One more quick question online. Do you think the migration is symmetrical?
I have no idea. And that's going to be a difficult question to answer. Like I mentioned, there's been some tagging studies done where they've tried to see and determine migration routes, but with such large populations, even when you tag them, the tagging return is really low. And there's been some evidence that maybe tags were collected across the equator, but there's even cases where those might get tossed out because people thought like, oh, this is an error. This didn't come from all the way over there. Yes. Yes, a lot of so all the samples we collected were through different collaborators, and that includes seafood processing plants, other researchers, local fishermen, and like recreational and commercial both. So there's been a lot of help from them as well. Oh, sorry. The question was if there has been any collaboration with fishers in the area for the sample collection.
Okay, great. Thank you. Oops, sorry. So our next student awardee is Claire Rosemond, who is a PhD student in fisheries, wildlife, and conservation sciences, advised by Dr. Scott Appel, received the 2020 McNeil Award, and we have a video.
Hi, everyone. Thanks for listening to my Ignite talk about marine heat wave impacts on black rockfish body condition. My name is Claire Rosemond. I'm a PhD candidate in the Department of Fisheries, Wildlife, and Conservation Sciences at Oregon State University. This project is part of my dissertation and was completed in collaboration with Kevin Martin, a visiting master's student from France, and Scarlett Arbuckle and Scott Appel at Oregon State University. My favorite motto is never swim alone. And I have followed that throughout this project by working with many OSU undergrads, fishing community members, tribal members, and even my family members. This project was made possible by their involvement and funding through OSU Hatfield and NOAA. I don't want to get too depressing here in the introduction, but thinking about how stress has affected my mental and physical health in the last few years has helped me frame my research questions about how stress may impact fish populations. I think about how external environmental factors might impact fish health. Recently, periods of unusually warm water or marine heat waves have occurred in the Pacific. These events are predicted to occur with greater intensity and frequency. This figure illustrates how, over various time lags, environmental stress impacts multiple levels of a biological system. My research aims to understand how anomalous conditions in the environment influence fish health or body condition, measured as lipid and muscle tissue. My research is focused on black rockfish off the central Oregon coast in nearshore habitat. Black rockfish are caught in recreational and commercial fisheries in California, Oregon, and Washington, and are the most commonly caught species of rockfish in the Oregon recreational fishery. We collected muscle tissue samples from black rockfish caught on recreational fishing charters out of Newport, Oregon, from 2015 to 2020. Sample collection gave us a chance to interact with the public, fishing industry, and management agencies. Using proximate analysis, I measured the lipid fraction of black rockfish muscle tissue. The muscle tissue samples were dried and ground, and lipid was extracted using a combination of solvents. A total of 1,654 samples were processed. After deciding on how to measure a potential response, I needed to think about which oceanographic conditions might influence food web dynamics, productivity, and temperature in this region that might subsequently influence fish health. The El Nino Southern Oscillation and the Pacific Decadal Oscillation are large-scale drivers of both ocean productivity and sea surface temperature in this region. These graphs show ENSO on the left and PDO on the right temperature anomalies over the time frame of this study. A major driver of primary productivity in the region is upwelling. Phytoplankton and zooplankton are the base of the marine food web. Southern species of copepods are generally more lipid poor than northern species and were more prevalent during marine heat waves. Over the time frame of this study, 2 marine heat waves occurred, one from 2014 through 2016 and one in 2019. These graphs of sea surface temperature highlight that warm water moved near shore during the marine heat waves. I used generalized additive models with muscle tissue lipid fraction as the response variable and local and regional environmental drivers at various time lags as the predictor variables to explore how oceanographic conditions impact fish body condition. Now, I will go over some results from the generalized additive models. I found that as upwelling, measured as QD, the coastal upwelling transport index, increased, there was a positive response in body condition. This effect was stronger in males. I found that as the southern copepod biomass anomaly increased, remember southern copepods are lipid poor and were more prevalent during marine heat waves, there was a negative response in body condition. This effect was stronger in females. The influence of sea surface temperature was weaker, but for females, I found an increase in body condition up to around 12 degrees and then a slight decrease. And for males, I found a slight decrease as temperature increased. I've learned that some important environmental drivers of body condition are upwelling, the copepod community, and local sea surface temperature. I've also learned that under stressful conditions, we, the fish and us, have some ability to adapt and survive. My next steps are to investigate how marine heat waves impact black rockfish reproductive development. I'll be interested to see if there are any shifts in spawning phenology or increased rates of skip spawning during marine heat wave years. Thank you to the Hatfield community and donors for making this research possible.
Claire, are you willing to answer some questions? You are online with us. You can go ahead and put it in the chat. Claire says yes. So if there are any questions, she's willing to answer via chat.
Yeah, I would just like to say that I really enjoyed your presentation, and I just wanted to ask if you've thought about how the models that you're creating from this research with black rockfish plays into other species of rockfish that are found off of our coast. Like, is there any connection between other species of rockfish, or is this significant to just black rockfish?
And there will be an awkward pause as Claire types her response. All right, I'll work on getting an answer to that question as we move forward. Go ahead, Ichan. Oh, I've got the answer. Possibly similar responses to other rockfish with similar life history, strategies, and habitat. Thank you, Claire.
Okay, all types of different ways of interacting. Our next speaker and awardee is Dr. Dawn Barlow, who received her PhD just recently in Fisheries, Wildlife, and Conservation Sciences. Her advisor, Lee Torres from the Marine Mammal Institute, she received the 2021 McNeil Award.
Thanks, Isabella. Ready.
All right, thank you all for being here. I'm excited to share just a little slice of the fourth chapter of my PhD, and that was conducted in collaboration with the co-authors listed here, and thanks to the generous support of the Hatfield McNeil Scholarship. So here we are in Oregon, but we're going to take a jump across the Pacific to Aotearoa, New Zealand, which was the focus of my PhD research, specifically this area called the South Taranaki Bight, which lies between the North and South Islands of New Zealand. This area is unique. It supports this cold water plume driven by a wind-driven upwelling system. This elevated productivity leads to aggregations in krill, which is the prey source for blue whales. So blue whales are the focus of my PhD research, and they spend just a little bit of their time at the surface when they're visible. And from times when we were able to observe them in the summertime, we've developed an understanding of their habitat use patterns, what drives their presence in this area. But there's a lot of times when we can't observe them. And a really effective way to study them when we can't be there is to listen. Sound is really important in the marine environment. It's how marine mammals communicate, navigate, and it's also how they perceive their environment, including things like pressures or disturbances from man-made noise sources. So we listened to this area using these marine autonomous recording units, essentially these fancy underwater microphones that we send to the bottom, and they were deployed in these 5 locations through the study area, and they recorded there for 2 years between January 2016 and February 2018. Another important thing to know is that different blue whale populations sing different songs, much like different human populations speak different languages or in different accents. And what you're seeing here are spectrograms. It's a visual representation of sound using frequency and time, and these spectrograms differ by different populations. And we wanted to know how many different populations overlap in our study area in New Zealand to get at what are the ecological implications of that overlap or separation between these different blue whale populations. We detected songs from 3 different populations, the New Zealand, Antarctic, and Australian blue whale populations. And we looked at the hourly presence of these different songs throughout our whole recording period over 2 years. And I'll show you some of these results here. And to orient you, each of these rows corresponds to a different recording location on the map. And number of hours per day will be that x-axis over time on the y-axis over time on the x-axis. So to start, this is the presence of the New Zealand blue whale population, and they are present all the time. They have some seasonal cycle in their singing, but on just about every day of our 2-year recording period, we detected this New Zealand blue whale song. This is in contrast to the Antarctic blue whale song, which we see in these 2 distinct seasonal peaks, kind of on the shoulders of the austral winter time. And then on a very rare occasion, we detected just during a short period at the most offshore westernmost hydrophones, these instances of this Australian blue whale song, a very rare occasion of maybe just a whale swimming by and hanging out in that area for one period. So what does this mean? Let's put these findings from our specific study area into kind of a broader geographic context of what's known about blue whales in the Southwest Pacific Ocean. We know now that from these acoustic recordings that the New Zealand song is detected in New Zealand year-round, and that there's year-round likely resident presence of this population in our study area. This is in contrast to very rare detections of this song in areas like Eastern Australia and only occasionally in Tonga. Antarctic blue whales migrate between feeding grounds around the Antarctic and wintering grounds at lower latitudes. So these 2 seasonal peaks that we were able to observe likely represent the northbound and southbound migration of this population. The Australian blue whale population feeds in the south of Australia in this area called the Bonnie Upwelling, and then they migrate west to Indonesia, where their breeding grounds likely are. And so our rare detection is perhaps more of an exception or a vagrant occurrence of an example of this blue whale that went a different way. So our study area is a primary habitat for this New Zealand population, in contrast to a migratory region or a vagrant occurrence of the Antarctic and Australian populations. And the reason this is important to understand is by understanding the timing and occurrence patterns of these different populations, we can think about their different exposures to threats from anthropogenic pressures. And so with that, I want to thank the many funders and collaborators and supporters that made this work possible. And in particular, thank you to the Hatfield Marine Science Center McNeil Scholarship that supported a lot of this work. Thank you very much. If there's time or there are questions, I'm happy to answer them. Yes.
Thank you. I really enjoyed your talk. I was curious with your hydrophones, are these specifically like attuned to picking up blue whale songs? Or are you getting everything and needing to sort through a whole bunch of noise to find the blue whales?
So these hydrophones, they're low frequency sensors. So their sensitivity, their best kind of sensitivity range runs up to about 800 hertz. So they are meant to record low frequency sound. So marine mammals like odontocetes, like dolphins and things that have higher clicks and whistles, we likely wouldn't pick them up, but they're really well suited for baleen whale vocalizations and also getting a sense of that low frequency noise band. Anyone else? Michael?
So is it live?
Yes.
It was really intriguing that, your rare occasion of an Australian one that went the other way. Is that the first time ever observed?
Yeah, as far as we know. I mean, so it's actually what's more surprising that how to phrase this? It's less surprising that we found it than it is that we didn't detect them more often. Because for a blue whale to swim between that feeding ground in Australia and the feeding ground in New Zealand is nothing. They could very easily get between the locations. So the fact that we don't detect songs from these other populations, particularly the Australian one, more often is really interesting to see that there is some sort of boundary or barrier between these populations. And there's always going to be an exception to the rule, right? And so this is an interesting example of they can get there and usually they're not there. They can get there, and when they're there, we can pick them up.
Hello? Okay. Which blue whale population sings your favorite song?
Oh, that's a great question. The Antarctic ones, they call them Z calls or Z calls, depending on in what part of the world you're in, and they make this cool Z or Z pattern in their spectrogram. And an interesting thing is that you can't actually hear them unless you speed them up to about 10 times the speed, because they vocalize at a frequency below what we can hear. So in order for us to hear them, you have to speed them up.
At the top. Get my steps in. Is there like a running hypothesis on the explained this, like that the vagrants, the ones that are outside their normal range, why? And I guess a follow-up of that is, do other populations of blue whales exhibit this, like, I don't know, the idea of just veering out of their normal expected ranges?
You are beautifully preempting a working paper that a whole bunch of collaborators are putting together, these rare vagrant occurrences. So they certainly can't, these are the largest animals on Earth and are really built for dynamic, like high, efficient movement. So they certainly can cover a lot of ground. And there are these rare occasions that may not be all that rare where one explores somewhere different. So for example, the Australian song has been detected off the coast of Chile. That's a long way to travel. And so there are these instances where every now and then there's a rare occurrence. And in terms of why, we don't really know. We're trying to piece that together by pulling together these examples.
All right. Our last and final speaker and student awardee is Olivia Boysen, master's student in Fisheries Wildlife Conservation Sciences, advised by Scott Happel. The award is the 2021 WIC Award, which is the William Q. WIC Marine Fisheries Award. It's intended to encourage graduate student research in the area of marine fisheries ecology with special interest in Pacific whiting, or intend to fund graduate research in marine fisheries and ocean-related research.
Yeah, sorry, can everyone hear me with this? I'm recovering from COVID, so bear with me. All right. Hi, everyone. My name is Olivia Boysen, and I'm a first-year master's student advised by Dr. Scott Happel and Dr. Suzanne Brander. And today I'm going to be talking about my project on myctophids as biotransporters of microplastics to mesopelagic food webs. So microplastics are classified as fibers, films, foams, and fragments less than 5 millimeters across, which have been found in waterways and oceans worldwide, and we are just now beginning to understand the effects plastics are having on marine organisms. So at the individual level, consuming microplastics can cause gut impaction and reduced ability to feed. Plastics also absorb chemicals from seawater and can transfer those to the organism, causing them harm such as reduced fertility and poisoning. Little is known about the microplastic levels in pelagic fishes, particularly those that live in deeper waters like myctophids. So myctophids are a family of lantern fishes and are likely the most numerous vertebrates on the planet, and therefore are hugely important to marine food webs. Myctophids perform diel vertical migrations, meaning during the day they stay about 1 kilometer deep in the water column, but then at night they swim to the surface to feed. They do this to avoid predation, and also they're following their food source, which is mainly krill. It's at the surface where microplastics are likely concentrated because of their buoyancy, and this is where myctophids are likely consuming plastic directly by mistaking particles as food or indirectly through contaminated zooplankton. With these plastics retained in their guts, they migrate back down to the deep where they are key prey items for many species, including whales, squids, and larger fishes. Additionally, their fecal pellets also sink to the benthos, likely containing the egested plastic. Thus, myctophids may be serving as a vector for transferring plastics from the surface to their predators in the deep sea, which has huge implications for marine ecosystem health and more broadly human health. Because myctophids are so prevalent, huge quantities exist in museum collections, and for my thesis, I'll be investigating the rise of plastic ingestion over time by looking at archived myctophids from the 1960s to the present. Plastics were first manufactured in the 1960s, and since global production has continued to increase. I expect to find that the rate of plastic consumption by myctophids exhibits similar trends, but with a time lag of several years, for larger plastics to degrade into smaller particles. The second chapter of my thesis will be using myctophids collected at varying distances from the Columbia River mouth, as plastics may be coming in from river systems into the ocean. The processing of these samples from both my chapters will follow methods standard in the microplastics field. These steps include dissection, digestion, filtration, filter picking, and analysis with an FTIR. First, each fish is dissected and their guts are removed and placed in a vial with 20% potassium hydroxide. This chemically breaks down the organic tissue, ideally just leaving the plastics for the next step, which is filtration. The contents of these vials are rinsed into a vacuum filter setup containing filter paper, which is then removed with those plastics on top. And each of these filters will be looked at under a stereoscope, and all these plastics will be photographed, and then later they'll be analyzed with an FTIR. So spectra from this machine will be compared to a known database, and so we can determine the polymer type, and we can see if these plastics are anthropogenic in origin. To avoid modern contamination, all this work is performed under a flow hood used with a HEPA filter. And we also take air and potassium hydroxide blanks to account for background contamination during all of these processing steps. So this project provides a unique way to utilize museum specimen to help answer questions about myctophids, but ultimately allows us to better understand the impact of future scenarios of pollution if we continue to produce plastics. The funding from my Hatfield Award allowed me to purchase the supplies needed for this project and also to visit the marine vertebrate collection in San Diego, where I was able to process samples for my time series chapter. And my next step is to visit the Burke Museum to get the remaining samples for this study. So the microplastics lab has recently moved to the new Marine Studies building, where I'll be continuing this work, and I'm excited to see the results from this analysis. Yeah, and with that, I'd like to say a huge thank you to my committee for all their help with this project, and to the Bill Wick Marine Fisheries Award for the funding. And I'd also like to thank the folks at Scripps for allowing me to use their specimen, and thank you all for listening. You can take any questions. All right.
I'm wondering how much variability there is in the amount of myctophids caught throughout the years. It's amazing that you have such a large data set, such a long-term data set. Are there any gaps in your data set where there are years that there are no myctophids or no samples that you can process, or is it pretty consistent?
Yeah, that is a challenge of these kinds of projects. That's why I'm using 3 different collections for this work. So I recently visited, the Scripps Marine Vertebrate Collection, where I got about 120 fish for this study. But to fill in those time gaps where they weren't collecting as many down at near San Diego, I'll be going to the University of Washington Burke Museum, and they also have kind of to fill in that time frame. They have collections that were mostly done in the 80s that I'm missing right now. And then also the University of Oregon has an ichthyology collection, which I'll be using some of those as well. Yeah. Right. Yeah, so the question was, how do I control for contamination from the nets? Yeah, so this is difficult because at the time they were collected, obviously the people didn't know that they'll be looking at plastics. So the best we can do is kind of to take background from the liquid that they're stored in. And also there were a few studies to see if they were, quote, net feeding, so if they were consuming things while being dragged through the net. And they didn't find much evidence of that occurring. So when they're caught, usually they die pretty quickly if they're not already when they're on board. And so, yeah, that is something to consider. Yeah. Oh, right. Yeah. Yeah, that's also something we aren't really able to detect, because these collections happened so long ago. But yeah, in the more recent studies, there's a couple collections happening this year. We have more background contamination and things like that we'll be using to compare with those. Yeah. Okay. Right. Yeah. Yeah. So the question was, how am I going to account for the varying locations that they were sampled from? Is that correct? Yeah. Yeah. So that's kind of tying in chapter 2 of my thesis, where I'll be looking at to see if distance kind of from shore, or distance from maybe a city or a plastic source actually affects, the fish that are further out. But again, yeah, without those like historical water samples, it's really hard to do that. But I'll be using kind of their coordinates where they're caught to take like a distance from that to shore, if that makes sense. And so I'll kind of be comparing ones that are caught closer to shore to ones that are caught further offshore, just by using that data that came with the samples. Yeah. Mm-hmm. Mm-hmm. Yeah. Yeah, so the question was, am I going to be looking for plastics in other areas of the fish, not just their guts? And yeah, that's actually a part of my thesis that might get pushed to a PhD project, is to look and see if there's translocation, is what it's called, when those fibers or particles move past the digestive tract and into other tissues like the liver. And there's been some studies that are showing that in anchovy and smaller pelagic fishes similar to myctophids, that these plastics are able to move outside that gut. And so for those, I'll be looking at different areas of the body, like their livers, also potential for like nanoplastics to enter their brains, and also their gills is another thing I'll separate out and see if we're finding those there as well. Yeah, good question. Mm-hmm.
Okay, well, so I guess this is a moment of stating just how diverse our projects are here, and how diverse marine science is, and the sheer range of projects put together and worked on by our faculty, our graduate students, and our undergraduates. And that really is the heart of the Hatfield Marine Science Center, having all these complex collaborative efforts doing projects located in the marine environment. And really, it supports our mission to improve scientific understanding of marine systems, coastal processes, and the resources, and how those apply to the knowledge of the social, economic, and environmental issues of our region, as well as globally. The student awards and the scholarships directly support our students as they pursue their research goals. They also recognize and again validate the outstanding research efforts of our students. Thank you for joining us today. Please join me in congratulating the awardees. Just a special acknowledgement and recognition. We want to acknowledge Shannon Reed, Cinnamon Moffitt, Melissa Reith, Colette Marco Nelson, and Tammy O'Connor for making this year's symposium a success. Thank you all for coming. And Cinnamon's reminding me to remind all of you, turn in your ballots. please submit your ballots in our ballot box. We'd like to invite all the awardees, their advisors, mentors, and special guests, and summer interns to dinner in the second floor mezzanine. And a reminder, again, please submit your ballots. Thank you for all joining us.